Hello, I'm Elisa Manzotti, CEO and founder of Cell and Gene Therapy Insights, and a really warm welcome to our latest webinar on GMP raw materials for cell therapy manufacture. So the cell and gene therapy industry is undoubtedly entering an exciting phase of accelerated growth, which carries with it many logistical challenges right from the very start of the supply chain. This webinar focuses on the critical issue of raw materials, and it forms part of our forthcoming spotlight on this very topic, which will be published next month. So this webinar provides an opportunity to hear from three leading experts who will be joined by our guest moderator today, Jean Stanton, who's also guest editor of our Raw Materials Spotlight. Following a 10-minute presentation by one of our speakers, Jean will be grilling our panelists with a selection of questions. And of course, as always, we would love to hear from our audience members too. If you would like to pose a question to our panelists, simply pop your question in the Ask a Question box under the webinar viewing pane, and we'll do our very best to respond. So I would now like to introduce our guest moderator who will be running today's session, Jean Stanton, Director of Regulatory Compliance at Johnson & Johnson. So Jean joined Johnson & Johnson in 2008 within the Pharmaceutical Quality Assurance Organization after more than 20 years in the healthcare industry at a university-based medical institution developing cell-based therapies. Her current responsibilities are the deployment and maintenance of the R&D compliance program for all GMP aspects within J&J's pharmaceutical sector, which includes the development of the quality and compliance strategies to support all new platforms that are in-licensed or developed within the pharmaceutical sector. So Jean, thank you very much for joining us today and for kindly moderating this webinar. I'd now like to hand over to you to introduce our panelists today.
Thank you. I'm delighted to introduce our expert panelists for today's webinar. Firstly, we have John Tomtishen, who is the U.S. Product Steward at Novartis Pharmaceuticals within Cell and Gene Technical Development and Manufacturing. In his role, John owns and manages the process knowledge for cell and gene therapy products for late-stage clinical development through commercial lifecycle management activities. He serves as a single point of contact within the Novartis network to ensure a seamless flow of knowledge and information across the organization. Today, he is joined by Dr. Diane Wotta, Senior Director for Quality and Regulatory Affairs in R&D Systems, Inc., a Bio-Techne brand. Diane is the Senior Director of Quality Control, Quality Assurance, and Regulatory Affairs at R&D System, which offers approximately 350 FDA-regulated products through both diagnostic and the biotechnology divisions. Her 20-plus years tenure as R&D Systems has provided her with extensive experience working in the science-driven, FDA-regulated environment. And our third panelist today is Dr. Sadik Kassim, Vice President, Process and Analytical Development for Mustang Bio, Inc. It is a clinical stage biotech company focused on the development of novel CAR-T therapies in partnership with the City of Hope National Medical Center and the Fred Hutchinson Cancer Research Center. Previously, Sadik was head of analytical development for the Cell and Gene Therapy Unit at Novartis Pharmaceuticals, where he contributed to the BLA filing of Kymriah for pediatric ALL. So what we'd like to do today before getting into the Q&A section of our webinar, Diane will take us through a short presentation to outline some of the key issues surrounding quality requirements for raw materials. So Diane, over to you.
Thank you, Jean. I'll start today with a short presentation. First, I would like to say hello to all the participants online for this webinar on GMP raw materials. I would like to thank Elisa Manzotti for hosting the panel on such an important topic for emerging medical products, as well as all of the other panelists. Again, I'm Dr. Diane Wotta. I have a PhD in pharmacology from the University of Minnesota here in the United States, and I've been working at Bio-Techne for 20 years. In 2002, I began working with pharma companies either in cell culture and fermentation for raw materials or in quality control QA/RA for in vitro diagnostics and reagents, as well as raw materials or ancillary materials for medicinal products. From this slide, you can see that Bio-Techne has over 200 scientists out of the more than 1,800 employees, and my team consists of over 90 quality and regulatory affairs personnel. We have been manufacturing recombinant protein products since 1989, as well as other products such as antibodies and kit products in both research and IVD grades. Bio-Techne at this time has 3 divisions: the Biotech Division, Diagnostics Division, and Protein Platform, which is our instrument division. I believe that you would all agree that the landscape for raw materials as well as their resulting medicinal products is complicated. And as you move from country to country, even the naming or categorization of the final medicinal products can change. As an example, in the EU, advanced therapy medicinal products, or ATMPs, are classified into 4 product types: gene therapy, somatic cell therapy, tissue-engineered products, and combined tissue or cell products associated with the device. In the United States, the definition is more broad than in the EU, with only 2 classifications: cellular therapy and gene therapy. The cellular therapies themselves are complex due to their biological nature, and the ancillary materials or raw materials that are used to manufacture these cellular therapies, or ATMPs, are also manufactured in biological systems. This only compounds the variability of the final medicinal product manufacturing process. This complexity of the biological systems leaves the ATMP regulatory environment and the raw materials a complex and dynamic environment. The regulatory framework provides for safety, identity, purity, potency, and clinical efficacy of the resulting ATMP. The majority of these requirements can be tested, but there are unknown risks, especially if the raw materials are manufactured in the presence of animal-based materials. The regulations listed for raw materials or ancillary materials are for Europe, Japan, and the United States, as well as some international guidelines. However, these can still vary depending on the country where the final product will be manufactured and utilized. Chapter 1043 of the United States Pharmacopoeia, along with the European Pharmacopoeia 5.2.12, and the WHO and ICH international guidelines, these provide guidelines for developing appropriate raw material qualifications. Such programs should focus in typically 6 areas: the identification of the raw material, selection and suitability for use in manufacturing, characterization, vendor qualification, quality assurance and control, and lastly is risk. So I'd like to focus on risk for a minute. The level of risk associated with an ancillary material will affect the qualification activities that are necessary. USP 1043 provides a framework for classifying ancillary materials into 4 different tiers based on risk. Tier 1 would be low risk, highly qualified material or pharmaceutical grade material that would have a DMF on file, and qualification on the part of the manufacturer would be minimal. Tier 2 are well-characterized materials manufactured under a certified quality system with specifications for the final raw material clearly defined. This level has typically been manufactured for the cell and gene therapy world, and most of the animal-containing reagents should be removed from the manufacturing process, if possible. Tier 3 and Tier 4 would be higher risk materials that may be manufactured in a pilot stage laboratory or an academic laboratory, and they're not typically intended for cell and gene therapy use. Finalizing GMP requirements and quality standards for the development, manufacture, and clinical testing of ATMPs in the EU and the US has been a difficult task, which arises from the lack of harmonization of the way GMP standards are applied. Here is a listing of raw materials that may be used in the ATMP manufacturing process. Depending on the process being developed, there is a potential to require the utilization of several of these raw materials in a single ATMP manufacturing process. However, a qualification and risk assessment should be performed to minimize the risk regardless of the quality of the raw material. The development of medicinal products begins with the completion of the preclinical proof of concept studies through to clinical trials. Raw material quality is important to consider throughout the manufacturing process. Process changes at later stages entail risk, requiring validation and comparability studies, whereas the earlier you incorporate the final raw material that will be used in your process, the less work will be needed to revalidate these changes. I've included a depiction of the medicinal product pathway through the regulatory process as referenced in the paper by Datella and Lodge, written and published in Cell and Gene Therapy Insights in 2016. ATMP developers will be able to use the lower grade of raw materials for non-clinical phase 1 and into phase 2 clinical trials. However, GMP-grade materials will be needed if they are available when entering into phase 3 clinical trials. Raw material changes between these two stages will take time and effort to revalidate. The quality of the raw material plays a major role in the safety and efficacy profile of the final ATMP. Variability in raw materials will be minimized by using a higher grade of material early in the developmental process. Additionally, you will want to secure the supply of your raw material with your supplier and provide your specifications if they differ from those that are included on the supplier's certificate of analysis. Validation and comparability are needed at later stages when a change in raw materials is initiated, versus only a validation would be required at the early stages to convert from a research-grade raw material to a GMP grade. Critical performance specifications for ancillary materials include safety, identity, purity, potency, bioactivity, and of course, reproducibility. Testing requirements are listed for animal-free proteins, which are considered Tier 2 ancillary materials. If using animal-containing raw materials, additional testing is required, and those tests are listed in this slide. In summary, I'd just like to make a few points. Country-specific regulations need to be considered when manufacturing or qualifying a raw material. Safety is paramount. Reproducibility is critical to the manufacturing process, and regulatory support from your supplier will be needed as the ATMP moves through the various clinical stages. Also, some raw materials may not be currently available in GMP grade, and it is best to contact your supplier early in your developmental process to discuss the possibility of getting those raw materials manufactured into a GMP grade. And finally, I'd just like to say that R&D Systems has custom GMP manufacturing capabilities for proteins, antibodies, and media. And we also would like to, I'm pleased to introduce the Trackless GMP small molecules. That will be coming soon. Thank you. Back over to Jean.
Thank you, Diane, for your presentation. I think this raises a critical issue within our field. How much confusion there is regarding the terminology being used for our raw material quality and the impact it's having. And I guess I'd like to start with Sadik to get your input on what you've seen as some of the problems we're facing and its impact in your experience, both, with Novartis, at Mustang Bio, and some of your other experiences.
Yeah, thanks, Jean. So first, thanks, Diane, for providing that really comprehensive and I think informative overview. I think, ultimately with raw materials, my understanding and our approach has always been that ultimately the sponsor or the user is ultimately responsible for the quality of the final cell therapy product. And within that context, also responsible for the quality of the ancillary material. A lot of these suppliers of the ancillary materials have, I would say, different labels, and different labels mean different things. So, for example, RUO may mean one thing in the hands of one supplier versus, CGMP in the hands of another supplier. So I think within that context, as a user, we always think in the same terms that Diane mentioned in the 4 levels of potential risk as per the USP guidance. And within that context, how can we, work with the supplier early on to figure out collectively, both as the sponsor and as the supplier of the ancillary material, figure out a low-risk strategy for qualifying the ancillary material to incorporate into our own trial. And I'll give you one case study. This is something that I was involved in while I was at the NIH early on when I was working with Dr. Steve Rosenberg. I was working on the CD19 CAR-T program that was eventually licensed to Kite. And, early on, Kite, and this was work spearheaded by Mark Better, who's currently at Kite Pharma. So Mark was the head of development at Kite, and I was working at the NIH. So I was the institution that was sending the manufacturing process to the industrial sponsor. When Kite came in and looked at the way that we were making the CAR-T cells, the CD19 CAR-T cells, they immediately identified, certain reagents that we were using in the academic manufacturing setting that would be problematic in the industrial sponsored clinical trial setting. One of those was, for example, we were using human AB serum. The work led by Mark Better, which he's published on, immediately said this is a potential liability for a multi-site clinical trial. So we need to figure out a replacement for this human AB serum. We need to figure out a replacement for some of these other additives that we were using in the process that weren't up to the standard. So early on, to Diane's point, the folks at Kite incorporated those changes, realizing that, incorporating the changes early in the clinical development cycle will save you time and resources down the line. They incorporated those changes and were able to really roll out, this multi-center trial. And I think it really comes down to the user and the sponsor, assessing the risk, seeing what are the major fires that need to be put out, and then, working with the supplier early on to come up with a reasonable qualification plan for using and incorporating those ancillary materials into your own program.
And I'd like to add a question on to that. John, we'll start with you. What are the major needs in terms of raw materials that currently cannot be sourced as GMP grade?
So that's one of the challenges that we face, right? Because not every material right now is GMP readily available. So it really depends on, to Sadik's points and stuff too, of really doing appropriate risk assessments to make sure that you focus first on raw materials that are considered higher risk. So one of the things that's well known within the cell and gene therapy industry is that there's a lot of variability between patient material, right? So one of the things that to differentiate between variability within your raw material from the patient is actually working on ensuring that you have the appropriate quality systems in place for other raw materials in your process. So anything around your vector, media, and things like that. And then this goes into also timing of when you make changes and stuff too, right? Because it depends on what your end goal is and different requirements for different clinical trials or proof of concept assays and stuff too. So if you make a change much later on in your process, it requires a lot more effort. So you need to have clearly defined comparability protocols and things like that.
Great, thank you. And I think on that line, and we could go back to Diane, if, there's a couple questions coming in from our attendees regarding, the differences between the EU and the US, those requirements for raw material programs. One of the questions talks about, what's required for these early phase trials, and then later on, as far as, going in phase 2, phase 3, and does the EU require that your raw material program be GMP earlier than the U.S., or is it the other way around?
Well, I would say that at this time, when you file, it's always best to do a pre-submission and to meet with your regulatory body. And then, as was stated, not all of the raw materials can be manufactured or are manufactured at this time in a GMP fashion. And so you do have to provide those risk assessments. So I wouldn't say one is requiring it more than, or I wouldn't say the EU is requiring it or more or sooner in your phases than any other regulatory body. But I think it really depends on how well you have characterized the raw material and how well you can provide that risk assessment to that regulatory body.
Yeah, and to just add on to that is, right, so the requirements are different globally as well, right? So it really depends from a company's perspective or academic institution perspective, it really depends on what your end goal is, right? So are you interested in some early stage clinical trials, or are you looking to do, say, focus on domestic manufacturing, or are you looking more at a global organization as well, which helps tie into the decision of when you make changes to the quality of your raw materials.
We are also getting some questions, and I'm going to ask Sadik to respond first, right? And it's regarding vector manufacturing, right? And how much clarity there is around the quality requirements. One of our attendees is asking that, often our clients ask for GMP-like or high-quality plasmids instead of full GMP. What is your opinion here? Will the authorities accept this GMP-light for critical starting materials, or do we need to look into, full GMP?
Yeah, it's a great question. And vector is an interesting ancillary material because, especially for ex vivo cell or gene therapy modification, it's a starting material, but then there's also starting materials required to make the vector. And I guess the questions here are asking about the plasmid. So, for lentiviral vectors, for example, most systems use a 3 plasmid or a 4 plasmid system. And the question becomes, the quality of the plasmid. So the plasmid is never going to, interact with the cells. It's rather just the raw material to make the viral vector. In the end, the viral vector is going to interact with the cell therapy drug product, but, the plasmids aren't going to interact. So within that context, what are the regulatory requirements? And, I've seen, in my experience, I've seen different sponsors approach it differently. So I know certain, there's academic groups, for example, that use research-grade plasmids to make their GMP viral vector. So the plasmid itself is research-grade, but the final process for making the viral vector is under full cGMP conditions. And they've, taken this type of ancillary material all the way to phase 1, phase 2 multi-center trials. And, within that context, I've seen industry sponsors as well maintain that similar workflow. So, using research-grade plasmid to make GMP viral vector that'll then enable a phase 1-2 clinical study. But I've seen the other end of the spectrum as well. I've seen from the beginning, especially if sponsors are thinking of going global with their ex vivo gene-modified cell therapy product, that they, given that there is a lack of coordination, it seems, in terms of guidances between the different regional and local authorities, they just from the beginning build for a higher, quote, quality plasmid. So they'll start off with the GMP quality plasmid with the idea that that would enable for the most seamless approval across the different regulatory bodies. So, getting back to John's point, I think within the U.S., from what I've seen, the FDA will accept, research-grade plasmid if the subsequent downstream activities related to GMP manufacturing of the vector all occur with the proper C of A's and chain of origin and all the other documentation. Whereas, if people are thinking more globally, they'll incorporate the higher quality level plasmids just with an understanding that it would enable for a more seamless approval process. So really, I think in the end, it's the clinical development strategy. If it's global versus local, and understanding what markets you want to roll your product out in, and developing your raw material strategy around that clinical development strategy.
Thank you. We have another question here that I'm finding quite interesting that I want to pose to all of the panelists. We hear that the regulators are looking for, the best case is pharma-grade raw materials. But what about when we're talking about raw materials or consumables that have 510K clearance? What are the regulators expecting? What level of qualification and or validation of that material is required?
So I can start with that. So if your raw material is already 510K cleared, then it will already be, and it would depend on what class it's for. If it's already 510K cleared for a, in vitro diagnostic, then it would still require more validation and requirements than other products. Sadik, do you have other comments on that?
Yeah, I guess with regard to qualification, to your point, Diane, it's really qualified to varying extent on the basis of, the risk mitigation plan and the nature of the ancillary material. And, in my experience, usually people qualify the ancillary material around 5 parameters. So, source, purity, identity, safety, and suitability. And the level of qualification will ultimately depend on the intended application. So, and again, this is where, having the sponsor and the user and the provider, the supplier and the supplier of the ancillary material and the sponsor of the clinical trial engaging in early conversation is really helpful to, appropriately develop a qualification plan for that specific ancillary material.
To Sadik's point as well, about risk and stuff. So it's important to have appropriate risk measurements in place, and depending on what stage you're in, the risk will be different for the quality of the material.
Yeah, exactly. And, I guess one point that I would ask Jean and maybe Diane as well, because Jean, I've noticed that you've talked about this issue with regard to ancillary materials and quality materials, something that I haven't really paid attention to, and I don't think many people pay attention to, but it's something that definitely one needs to keep in mind when putting together a qualification plan, is the issue of particulates in raw materials. What sort of advice and what sort of experience would you share with the group? How should we approach the issue of particulates in quality material in raw materials, and how should we incorporate the potential risk of particulates in terms of our qualification plan for the ancillary material?
That is a good question, and particulates are something that near and dear to me, and I've been working with and talking about for a while now. And I think the first thing developers need to do is start working with their suppliers. More and more, what I notice is that suppliers are much more willing to share what information they have around particulates. The regulatory expectation is that what's in your product. It's not, it's not nothing. We have cells there. It's hard to see what's in there, but knowing what's additive, what you can take out, and what you cannot take out is very important. And I cannot emphasize enough how critical it is to engage with the suppliers to have that information. And like I said, more and more what I'm seeing is they're much more willing to share what they have and then work with developers to identify particulates in their particulate loads if they don't already have that information.
Great, that's very helpful. And I guess, Diane, from the supplier perspective, like, how can, what can the potential sponsor of the clinical trial do to engage, most effectively with the supplier? Like, from your experience, what can sponsors do early on to really help facilitate the process with respect to ancillary material and raw materials, working with someone like you on the supply end?
So from the supply end, you would look at your materials as they're being manufactured, the raw material, that is, as it's being manufactured. And then it depends on how you manufacture, how you bottle, how the material is filtered in order to remove any type of particulates before the material actually leaves your facility. There are testing capabilities for particulates, but ideally you would not want to have particulates within your raw materials.
There's another question that I wanted to direct to both John and Sadik about how manufacturers are dealing with evaluating the effects of raw materials on their cells and the treatment or the effect they want to have. What assays are being used to evaluate those effects?
Well, that's a great question. I mean, I think with regard to the assays, it really all links back to the critical quality attributes of the drug product. I think one of the challenges with these autologous cell therapies, they're personalized. We don't have a full comprehensive understanding of the critical quality attributes. I mean, we understand some parameters that are important, like the viability of the cell, the product, for example, or the purity of the cell therapy product. But beyond that, with respect to phenotypic and functional markers, we don't have a full and comprehensive understanding. So I would say that that's one of the challenges with evaluating ancillary materials. When you evaluate the impact, you can evaluate the impact of using certain raw materials on these gross aspects like cell viability or purity. But, there may be other things that are measurable, but in the end, given that we don't really have a clear set of critical quality attributes, you always take a risk when you incorporate a new raw material because you may be measuring something, but it may not be relevant to the actual potency of the product. And, that's been a big challenge. I mean, John, how do you see it?
Yeah, so one of the other key components too is when evaluating anything, so looking at the impact of incorporating in raw materials on your cells is really also understanding the material of construction of any of the materials that you use in your manufacturing process. So making sure that you have a good understanding of what the components are made of. Are they compatible with the reagents and other things that you use? Because that can have a big impact on the overall final product quality of your cell therapy product.
So the next question I wanted to start by directing towards Diane, but then ask for John and Sadik to offer their insight as well. So do you feel there's sufficient regulatory guidance being provided to help clarify, around quality requirements for raw materials?
Well, it would be really nice to have guidance or a requirement that would be international, that everyone would acknowledge or recognize, but right now that's not available. So I think that the suppliers are trying to meet the requirements that are listed that I presented in the presentation as well, purity, identification, all of those physical requirements, but then leaving it up to risk, or going back to risk, the risk is really the animal components that would be used if there's any, and trying to remove them if possible. But then there's also the viral removal steps, virus of master cell banks, viral testing for adventitious agents. So those requirements, all of those requirements are really not clear, and it really depends on what country you're in. So I think coming up with a complete set of requirements would be beneficial for everyone, but it's just not there yet.
Yeah, I mean, I would agree with that. I mean, I think the, in terms of the requirements for the manufacture of the cell therapy product, the guidances are pretty clear, but in terms of the guidances for the manufacture of the ancillary material, I think the suppliers, for the most part, are doing a good job navigating, making these reagents in the absence of very clear guidances and how to make it. And, I think the challenge is, you look at the raw materials that are needed for making cell therapy products, they're pretty diverse. So I think maybe one of the struggles that the regulatory bodies are coming up with is how do you define, guidances for something as varied as, serum all the way to protein, cytokine growth factors to various, let's say, culture vessels. It may not be easy to come up with a comprehensive list of guidances for such a wide range of ancillary materials and raw materials.
And to that point, back to Sadik's point, the industry is still very young too, right? So the guidances aren't as clear as some of the more advanced guidances for other materials, or drug products. So monoclonal antibodies, vaccines, and things like that. So as the industry evolves, more guidances will be clearly defined, but as of now, there is a lack of clarity. But it goes back to Sadik's point that there is a wide variety of different strategies as well in on how to produce and manufacture cell and gene therapy products.
Thank you. Another question that has come up from our attendees relates to the cost, right? So when deciding to go with GMP, right, when working with clients, and I'll start with you, Diane, does cost tend to be one of the factors that drives that decision to shift from research grade to GMP grade in a product development?
So typically what I have seen is by the time we get contacted and people are interested, they've been using a research grade material. And by then, it's really time for them and not necessarily the cost of switching to the GMP grade. It's that they have a deadline or they're going to file by this date and they've already set their project is in motion and it needs to be filed by a certain date. And so getting them to step back and say, okay, now we want you to do a revalidation because we think it would be in your best interest to switch over to GMP materials at this stage. It'll be a lot easier for you in the long run. And we have a difficult time getting developers to take that step and to go back and maybe delay their project 6 to 9 months. So that's really what I see.
Yeah, and to add on to that, I think it goes back to some of the earlier points as well. So it really depends on what your end goal is, right? If you're an academic institution, it might be a lot different than if you're within the pharmaceutical industry, right? So from a cost perspective, it's much easier, and also from a regulatory perspective, it's much easier to make changes to raw materials earlier on in your life cycle of your cell and gene therapy product, because later on, as you change it, the regulatory requirements are much more stringent. So comparability assays, also additional validation runs and things like that. So it really depends the decision should really be based on what is your end goal for your institution or company.
Yeah, absolutely. And, within the academic setting, most academic centers are in the phase 1-2 setting. So, incorporating an amendment to an IND is fairly straightforward. But as you progress down the clinical development path, I mean, Tufts University had the study, which some people cite frequently, and I think it really speaks to the cost of implementing changes to something as basic as raw materials later rather than sooner. So, the Tufts study says that, the cost of an amendment for a phase 3 trial costs more than 3 times as much as an amendment for a phase 2 trial. So, you may be saving some upfront costs by using the less expensive material, but down the line, if you do switch over to the GMP grade material, it's not just the cost of the GMP material, but it's also, to John's point, the cost of the clinical comparability and the bridging strategy. And that's going to be much more costly as you progress down the clinical development path.
So I'm going to come back in this to managing risk, right? And this question was directed to John to start. We talk about risk-based approach when we're working with Europeans and managing risk. And what's your strategy for risk assessments, especially when raw material, it can be so dynamic? How do you plan that out?
Right. So it needs so in your risk assessment approach, you really need to prioritize where what materials or reagents in your process have the highest risk. So looking at it from both a supplier and quality perspective, reliability perspective, and things like that. So it needs to be holistic in your approach and how you evaluate the risk for different materials. So one material might be higher risk because of how it's manufactured and things like that. So that's you need to have appropriate risk mitigation plans in place as well. And it's much easier. And again, this goes back to defining your risk mitigation strategy. Really needs to be something that you consider upfront before you really change over or go with different materials in your process.
I think another question came in, and I would direct this to all 3 of our panelists. The EU GMP guidelines that were issued at the end of last year and go into effect in May of this year, how do you think it will impact just this idea of global harmonization and the movement towards that? Do you think that these guidelines could help be a driver?
Yeah, I mean, I think, given that we have at least, 2 approved gene therapy modified cell products, the Yescarta and Kymriah, and, in Europe, you have Strimvelis and some of the other products. As these products get approved, each of the regulatory authorities, they're learning from each other. They're understanding what it took to get, one product across the finish line in one geography versus another geography. And, it's the natural trajectory of this field is going to be consolidation. And these guidances are, one step towards that, hopefully inevitable path.
No, I have to agree with that. I think that having this defined in the EU will help in the United States as well and in other countries. I think that the definitions that are listed in the directive are the guidelines are very clear and will help everyone.
And to Sadik's point, though, too, I think the guidelines are clear now. But as more products are come to the market and stuff too, I think the guidelines will evolve and become even clearer down the road as well, as both in the EU and in the U.S.
Great, thank you. Very often people leverage experience and knowledge in raw materials for biopharmaceutical production on cell and gene therapy. Is there a situation that experience from the biopharmaceuticals would not apply for gene or cell therapy products?
Well, I think, the whole perspective of the way that raw materials are handled in just general drug development is very different from, the way that, it should be handled in cell therapies. And I don't have to tell this group, this group has experience, but, with regard to a traditional small molecule or a biologic, you make one large batch, and you have that batch where you can, which will undergo months of testing for particulates and residuals and all of these other components. And you have very clear analytical methods for determining the impact of various raw materials on the final impurities within the drug product. Whereas you don't really have that luxury with cell therapy products. Number one, these are biological products that have very short stability. So once you make a cell therapy product, there are some fresh cell therapy products. The Dendreon Provenge product, for example, had a shelf life of less than 24 hours. You're unable to really do the extensive testing on things like particulates and other contaminants. And within that context, that really impacts your raw material strategy because to measure the true impact of various raw materials on the final drug product, you don't have the luxury of having a large batch where you could do extensive battery of tests to measure the impact of various changes. So I think that there's always going to be that difference between the cell therapies, especially autologous cell therapies, and the traditional biopharmaceutical and pharmaceutical business. I hope that the guidances that are being put together will incorporate that nuance in terms of the nature of the scale of the products, in terms of how raw materials should be approached as well.
And additionally to that, I think that it would be, as the environment evolves, I think there will be new testing strategies that will come about. Companies will develop new testing strategies that could meet some of those timelines for these short-lived products. Hopefully that will be coming in the near future because I think that those types of testing capabilities would be really beneficial to the products.
Yeah, and to add to that, I would say one of the other interesting things would be as the industry evolves and things like that, so is from a patient's perspective as well, right? So right now, also taking into account patients, right? So these treatments are currently used really for really sick individuals and stuff, right? With refractory relapse, leukemia and things like that, right? So if the treatments become more frontline and stuff, where it's not necessarily as critical to infuse the patient as soon as possible, right? So the requirements or requests from might be different and stuff on the type of testing that you need to perform to release different materials and to really understand all the components and characterization activities as well. But again, this also relates back to the overall shelf life of the therapy product.
Thank you. This next question is to both John and Sadik. Is there any suggestion or thoughts around trying to identify secondary sources of ancillary materials to mitigate supply risk versus the impact on product quality?
Okay, yeah, so that goes back to the mitigation actions that you need to perform as well, right? Because you don't want to necessarily get into a situation where, say you have issues with a supplier or something for one of your critical raw materials. You want to ensure that you have appropriate substitutes as well that are comparable as well, so you can switch out, so you don't necessarily have an impact on your manufacturing and ability to deliver these products to patients.
Yeah, and within that context, if one has the ability to always think of backup strategies for critical raw materials, that should be part of the risk mitigation strategy. But, sometimes you don't actually have the luxury of something like that. I'll give you a very clear example. For CAR T therapies, people use various viral vectors to transduce or genetically modify the T cell. Some people use lentiviral vectors and other people use retrovirus vectors. With respect to retrovirus, the best way to enhance transduction efficiency or the overall genetic capacity is using this raw material called retronectin. The challenge with retronectin is that there's a supplier of that material. So it's critical to the overall process performance, but how do you incorporate a risk mitigation strategy for a raw material that only has one supplier? That's really a critical element. And, sometimes you don't have the luxury and you have to, make these decisions knowing the full risk of